Hello, my name is Igor Franović and I come from the Institute of Physics Belgrade in Serbia. So let me first thank the organizers for inviting me to hold the talk at this very nice conference. So my today's topic will concern some new insights we have recently gained concerning the impact of ketogenic diet in treating epileptic seizures using the framework of the next generation neural mass models. So if I were to convey the main message of this talk in a single sentence, I would probably say that we have discovered how the ketogenic diet gives rise to new types of adaptation at the level of single neuron dynamics, both the excitability adaptation and the spike frequency adaptation, which then translate at the level of collective dynamics by giving rise to new forms of multi-stability and transitions between the asynchronous and synchronous collective states. Now, bear in mind the point that the asynchronous states, especially with the low firing rate, are classically connected to normal, healthy, desired homeostatic states of neuronal populations, whereas the states with excessive synchrony are interpreted as related with epileptic seizures. So the results I will present have been obtained in collaboration with my friends and colleagues from the Ritten Center for Brain Science in Japan, in particular Dr. Sebastian Adam and Dr. Louis Kang. So let me briefly provide you with some background concerning the ketogenic diet. So ketogenic diet is a dietary treatment for epilepsy. Epilepsy is the third most prevalent neurological disorder, and we have known for years that it has a strong metabolic component, as do the Parkinson's disease or the Alzheimer's disease. Now, the main problem with epilepsy is about a third of patients remain resistant to classical medication therapies, the anticonvulsive medications. And then for these patients, some alternative therapies have to be developed. And in particular, one therapy of choice has been the ketogenic diet, which has a long history of application. It has first been applied in the 1920s, and it is still proven now to be very efficient, especially with children. Why with children? Because the children tolerate its administration much better than the older people. So ketogenic diet is actually a low carbohydrate dietary therapy, and it consists in the intake of fat versus carbohydrates and proteins in the ratio of 4:1. So it basically introduces a severe change of metabolism, and it drives the organism into the state called ketosis. So the ketosis is actually characterized by the switch in the main production mechanism of ATP, so the energy production mechanism. Instead of glycolysis, which is based on dissolution of the carbohydrates, we have a new mechanism of production of ATP, which is based on oxidation processes in mitochondria, which are then fueled by the ketone bodies produced by the liver by the solution of fatty acids, which derive, in fact, from the ketogenic diet. So why do we need to study the impact of ketogenic diet by different means? The point is that it is somehow targeting some metabolic pathways which are left untargeted by the conventional anticonvulsive drugs. So understanding its impact can actually help us develop some new forms of therapies, not only for epilepsy, but probably in the longer term for some other neurological disorders which have a strong metabolic component. And we hope actually to kind of foster a new direction of research toward metabolic therapeutic procedures for neurological disorders. So impact of ketogenic diet at the level of neuronal populations is manifested in a twofold way. One level is that the ketone bodies have an immediate impact on the excitatory communication between the neurons. By reducing such communication, they have an inhibitory effect on the excitatory synaptic channels, but this is not the main mechanism that we will focus on. We will actually focus on the other mechanism related to the activation of the ATP-dependent or gated potassium currents. So it turns out that the abolition of this glycolytic ATP production leads to the depletion of the ATP in the layer of cytoplasm very close to neuronal membrane, which in turn leads to activation of these outward fluxes of potassium, which have a hyperpolarizing effect, thereby reducing the membrane potential. So you can read more about this in some very important papers from the Gary Yellen group. So I will provide some brief background concerning the next generation neural mass models. They are a reduction approach, and they have so far pioneered actually by Montbriot, Pazzo, and Roxin. in this nice paper from 2015, and they have allowed us so far for a very systematic study of critical transitions, multi-stability, and control strategies. We have gained many insights concerning the interaction of populations, the impact of electrical and chemical synapses, stochastic and deterministic drive, quenched randomness, finite size fluctuations, emergence of bumps, waves, collective rhythms, deep brain stimulation in Parkinson's disease, working memory, different types of adaptation, both synaptic plasticity and spike frequency adaptation. So the reduction approach is based on the Lorentzian ansatz for quadratic integrated fire neurons and the equivalent Todd-Thomson ansatz for the theta neurons. So the reduction, its main advantage is that it is completely exact in the thermodynamic limits. So we obtain the low dimensional models which are amenable to bifurcation analysis, and then the population dynamics is characterized in terms of the mean membrane potential and the mean firing rate, but we also have insight at variance with the older generation neural mass models to the evolution of synchrony. So this can be done in two ways. One is, of course, to use the conformal mapping between the Kuramoto complex order parameter and the mean firing rate and the mean membrane potential. The other means is just the point that the onset of synchrony in neuronal populations is actually reflected in oscillations of the mean firing rate and the mean membrane potential as shown in this paper by Devalle, Roxin, and Montbriot. So I will briefly provide you with some insight concerning the main neuron model. The main neuron model we will consider is the quadratic integrated fire neuron. Of course, it has the normal form of the saddle node bifurcation, where this eta is the excitability of bifurcation parameter, but it is also endowed with an additional reset mechanism such that if the membrane potential exceeds a certain threshold, then its potential is reset to this value V reset, and this is considered a spiking event. So depending on the choice of this reset value, the quadratic integrated fire neuron model can fall to different classes of excitability, but the standard choice when this reset value is negative leads to class 1 excitability, where just the transition between the excitable regime and the regime of tonic spiking is mediated by the saddle node of infinite period bifurcation. So if the reset value and the peak value are chosen to be minus infinity and plus infinity respectively, then this model is also equivalent to the theta neuron model via this transformation. And of course, we know that the theta neuron model is a phase variable model. Its advantage for numerical simulation is that it has no reset mechanism. And in particular, we just consider as the spiking event once the phase exceeds the value of pi. So the main model that we will consider for the population dynamics is the population of globally coupled quadratic integrated fire neuron models with the metabolic feedback associated with the ketogenic diet. So the population is considered to be diverse in the sense that the local bifurcation parameters are distributed according to the Lorentzian distribution with delta and with the mean excitability parameter eta bar. This means that some of the units are excitable and some of the units are self-oscillatory. They're coupled they're coupled in an all-to-all way, such that we have chemical synapses between them. They're excitatory, they're characterized by the coupling strength K. And moreover, the synapses are assumed to be instantaneous, such that there is no finite time of processing of the incoming signals. Of course, this is a simplification, but a very useful one for analytical tractability. Then we have some impact of external stimulation, be it a constant one or the time-dependent one. And then the mean contribution from the metabolic feedback associated with the diet are these ATP-gated potassium hyperpolarizing currents. You see that these terms are local in the sense that they depend on the local membrane potential, but they have also a certain global component coming from the mean, or rather the population averaged ATP concentration. Of course, this is a simplifying assumption. There could be, of course, other models where this concentration of ATP could also be local. Here we assume that the dynamics of the ATP concentration depends on the production term, which is characterized by this finite time constant tau. There is a maximal also concentration that can be reached. Of course, the inverse of the production time constant is the production time rate, which can also be considered as a model parameter. And then the depletion of the ATP is due to the consumption of ATP by the neuronal activity. So you see that this term that decreases the ATP concentration is proportional to the population activity. Of course, using the transformation that I have described, we can also transform the original model of quadratic integrated fire neuron models quadratic integrated fire neurons to the theta population of theta neurons. Of course, one can follow the standard derivation of the mean field model using the continuity equation for the conditional probability density for neurons to have a potential V at moment t, given their excitability parameter eta, and then we assume that that the solutions of this equation generically follow the Lorentzian distribution characterized by the mean and the width, and you see that these parameters are actually these quantities are actually connected with the mean firing rate and the mean membrane potential, which ultimately leads to the next generation neural mass models for this type of a system, which at variance with standard classical neural mass models also has an additional equation for the dynamics of the ATP concentration. Of course, also note that one could, of course, consider this system as a slow fast system if we assumed that the production rate of the ATP is very slow, but here we do not assume that because we don't know a priori that such things occur. As the main control parameters, we will use the coupling strength K, the mean excitability eta, which is standard for populations with diversity, populations comprised of excitable and oscillatory units, and the main metabolic parameter will be the ATP production constant, or its inverse, the ATP production rate. So let me briefly explain the point on why the ATP-dependent potassium currents are considered to give rise to some form of adaptation at the level of single neuron dynamics. So here I have provided an example of a macroscopic stationary states. You see some small fluctuations arising from the finite size of the population, and the local structure of such stationary macroscopic state, macroscopic equilibrium, is characterized by the division of the population into a excitable subgroup and the oscillatory subgroup. And you see here at this figure the main impact of the ATP-dependent potassium currents. In particular, if there were no currents and no interaction between the units, you would see that this spike in frequency distribution of units would follow this red line, meaning that a huge part of the population would be excitable and a small portion of the units will be oscillatory. If the interactions are added, then the situation drastically changes. Most of the units are excitable and most of the units are actually oscillatory, given by this green line, and just a few units are excitable. However, if we add the ATP-dependent potassium currents, then the situation again changes drastically. Most of the population is still in the excitable regime, and the small part is oscillatory with frequencies less than those given by this green line. So also, if we take a look at the local ATP-dependent currents and their time averages given by this red line, and the variance is given by this blue line, we see that actually the negative contributions, which may be interpreted as, of course, the perturbations of the local bifurcation parameter, are such that the units closest to the excitability threshold have the largest contribution in the sense of being driven away from the excitability threshold, whereas also the units which are spiking have a negative contribution, meaning that the spiking frequency is reduced by the ATP-dependent currents. Of course, one can use the mean field model to construct the bifurcation diagrams, and it turns out that this structure of the bifurcation diagram drastically depends on the coupling strength K, gaining complexity for stronger coupling. In particular, for lower coupling, we just have a small region of bistability confined by the saddle node curves, which meet at the cusp point. And here we have the coexistence between the lower and the higher activity asynchronous states, and only the higher activity asynchronous states can undergo a supercritical transition via supercritical Hopf bifurcation to synchronous. So only this small portion of the phase space allows for the epileptic seizures. So of course, the transition can be induced by increasing the ATP production time constant, or interpreted in another way, the reduction of the ATP production rate. However, the transition to asynchronous states is re-entrant such that for higher ATP production time constant, we have the regaining of the asynchronous state. So for the higher coupling strength, the diagram is completely different. So we see that there is a larger area of bistability, but moreover, in this region, there are two codimension 2 Bogdanov-Takens points, and also two codimension 2-fold homoclinic points, which are not shown, but from the Bogdanov, for simplicity just, but from these Bogdanov-Takens points, there also emerge branches of Hopf bifurcations, and on these Hopf bifurcations, we see the generalized Hopf points or boating points where these hot bifurcations change character from subcritical to supercritical, and you see that both the lower and the higher activity asynchronous states can undergo transitions to synchrony, but also by subcritical or the supercritical way. So the interesting point is what connects these two diagrams. So in terms that turns out that these diagrams are actually a result of the system being very close to a codimension 3 bifurcation, also induced by this coupling strength K, and this involves a degenerate Bogdanov-Takens point where just reducing the coupling strength, there is a collision and disappearance of these Bogdanov-Takens points such that the curves of saddle nodes and the hot bifurcations detach. So I will briefly provide you with some insights concerning the control strategies that can be devised for switching between the, especially switching between the coexisting asynchronous and synchronous states. So we know that in the vicinity of the subcritical hot bifurcation, in this diagram, there is also the fold of cycles bifurcation, and this allows for the coexistence between the asynchronous and synchronous states. And then one can have, in the vicinity of this subcritical hot bifurcation, the different hysteretic transitions between the asynchronous and synchronous states, induced in particular by parametric perturbation, this being the ATP production time constant. This can be done by sudden or here gradual changes of the ATP production time constant, where you see also the delayed bifurcation as the effect of the slow passage through the bifurcation threshold. There are, of course, other strategies that can be applied to switch between the asynchronous and synchronous states. In particular, there is an external stimulation current effect by the excitatory or the inhibitory pulse of finite width. And also, there could be ATP shocks, which can be interpreted as a form of dynamic perturbations, where we introduce a sudden external either increases or decreases of the ATP concentration level, such that we induce a transition from the asynchronous or to synchronous state or back. And it turns out that also the sensitivity of this synchronous state is quite large to the ATP shock. So the negative one can almost be instantaneous to switch back to the synchronous state. Of course, one can use the similar strategy for switching between the coexisting equilibria. Now, the interesting point, of course, related to these dynamic perturbations and also like all other hysteretic effects, is that they can be used for therapeutic purposes in an interesting way, because they provide a certain type of irreversibility of the transformation, such that if the system switches from the synchronous to the asynchronous state by some perturbation, then of course this can have a long-lasting therapeutic effect, because the hysteresis by itself implies irreversibility of the effect or the persistence of the therapeutic procedure in this interpretation. So coming to conclusions, we have understood that the ketogenic diet introduces a form of excitability and spike frequency adaptation, so form of kinetic adaptation to the local dynamics, which is an interesting insight for future studies of neurological disorders with a strong metabolic component. Also, we have seen that the bifurcation diagrams exhibit a strong sensitivity to the coupling strength. And moreover, we have seen that for the larger coupling strength, there are new types of multistability and transitions between the asynchronous and synchronous states, both via subcritical and supercritical hot bifurcations. And there is an interesting link concerning this different pictures that we obtained for the lower and the higher coupling strength, because this can also be somehow related to the effect of ketone bodies, because as I have said in the beginning, the ketone bodies also introduce a certain form of synaptic plasticity, and they reduce the excitatory communication pathways. This can be, of course, interpreted as the reduction of the excitatory coupling strength. And you can see that indeed, if the coupling strength is reduced, the portion of phase space available for the synchronous, being the epileptic seizures states, just reduces. Also, we have provided an idea for the 3 stimulation protocols to control the switches between the normal epileptic-like states, being the external perturbation, the parametric perturbation, and the dynamic perturbation. And of course, you can read about these points in this very nice paper we have published in Chaos this year. Concerning the outlook, I would also like to point out that the findings that there could be subcritical and supercritical transitions to synchrony could shed some light on the recently sparked debate concerning whether there are means to predict the epileptic seizures by observing the critical slowing down, with some findings suggesting that there is or there isn't this critical slowing down. But we know that the critical slowing down is typically manifested for the supercritical transitions, and given that there could be subcritical transitions as well, according to our study, then the absence of critical slowing down in certain instances can be attributed to this point. Also, we could consider some types of model improvements, in particular by including different compartmental models for different types of ATP production processes, and also we could include some sort of synaptic plasticity to describe the effect of ketone bodies. So now the final questions which I would like to address for the audience, in particular the experimental researchers. So we would like their help, in particular by running some, to ask for their help concerning some potential experiments that could explain how during the ketogenic diet the reversal potential of the ATP-dependent currents changes independence of the membrane potential, and in particular other metabolic factors. Also, we would like some advice concerning the interaction between the potassium ATP-dependent currents and other leaky currents and their dependence on glia. With this, I would like to thank you for your attention, and I would be happy to answer to all of your questions.